Alright guys, today we're going to talk about the topic of DNA mutations. And I know when you think of mutations, you probably think of superheroes, like Spider-Man's mutation of having all of those spider qualities, or like mutated animals and like real science fiction stuff like this deer that you see here in the back of this truck. But I do want you to realize that mutations are actually what gives us as humans and other organisms variety. Okay, so all of these things that you see on the screen right now are actually because of a mutation. And in some instances, to me, it makes the organism more beautiful. Like these tulips here, they're called Rembrandt tulips. They're beautiful to me. So the mutation didn't harm them, it just made them different. Okay, the fact that you might have free earlobes and I have attached earlobes, obviously that doesn't change. It's not a negative effect, it's just a difference. So this here, two different eye colors, is called heterochronia. It's just different. Okay, so a mutation is actually what gives us a lot of our variety. Okay, so it's not always doom and gloom and like crazy stuff like you're thinking. Okay? Alright, so what I want to go over first is what is a mutation. So a mutation occurs anytime the DNA sequence itself changes. So even if one T becomes an A, that's a mutation. Now whether or not it's harmful or has no effect is something else. Okay? So if a mutation actually happens in a body cell, then it would only affect the individual. Wow, couldn't spell that for a minute. Okay, so as an example, if I decide to be stupid and go lay in a tanning bed every day for the rest of my life, that would affect me. Okay, now if there is a mutation in a reproductive cell, then obviously it could affect future offspring. Okay, so that's one of the reasons they actually do all that protection when they give you x-rays, right? That big heavy vest sometimes and all that. Okay, so the actual things that cause mutations are called mutagens. Okay, so some examples of things like that are x-rays, the sun's UV rays, right? Skin cancer and premature aging are big problems nowadays. There are chemicals such as asbestos, any kind of radiation going into space or anything like that. All kinds of things cause cancer and mutations nowadays. So radiation and chemicals actually have so much energy in them that they can blast your DNA into pieces. Okay? So like I mentioned a second ago, the most common effect of mutations nowadays, of course, is cancer. Okay? Alright, so let's talk about mutations, the two kinds of mutations that can happen on the DNA strand itself. Okay? The first one is called a point mutation, and all that means is one single base pair is changed. Okay, so like if you look at our example here, right, here's our point mutation. The T became an A, and that one single nitrogen base change caused this person to actually have sickle cell. Okay, so if it makes a new protein, then obviously it was harmful. If it's not affected, then it would be it would have no effect if the protein made is still the same. Okay, so let's look at this example with a normal sentence. The cat ate the rat. If we do a point mutation and change this one letter, the cat ate the rag. That gives that sentence a whole new meaning. Okay, and then sometimes it may not be affected at all. If you look up AGU, I believe you get serine. Let me make sure. Yep. And then if the U was replaced with a C and now I get AGC, I still end up with serine. So in this case, was the DNA mutated? Yes. Did it have a harmful effect? The answer then would be no. Okay? Alright, now let's do one that's a little bit more complicated and more deadly. These are called frame shift mutations. This is when it affects a single base, but instead of it being just changed to another base, it is actually deleted completely or added. And then that makes the entire strand behind that shift. That's why it's called a frame shift. Okay, so let's use this same sentence again. The cat ate the rat. If we delete this T, Okay, you guys know how DNA and RNA works. It works on a triplet code, right? So based on threes. So those letters are going to shift one. So the A moves over where the T was, and then that causes everything to move over. So if you notice, we're actually missing a codon here, okay? And then all of these other ones are called nonsense, okay? Because they are completely different than the original strand. So I want you to think of it like this, and I did this one time when I was taking the SAT, I think. So if you're taking a bubble test, right, and you're doing 1, 2, 3, 4, and you get down to number 5, and you're like, I'm not sure about number 5, let me mark it. So then you go to number 6, and you know the answer, but you accidentally put the answer to number 6 in number 5. That means everything from that point on has the possibility of being wrong, even if you know all the answers. Okay, so that's a good common example of a frame shift mutation. Okay? All right, so now let's scoot on to mutations that can actually happen on the chromosome itself. So these are obviously can be worse depending on how large the mistake is. Okay? So these occur in all living things, especially plants. Okay? And one of the most common ones is called a nondisjunction. And to me, y'all, that's a crazy word. You know a junction is where two things come together. And then a disjunction would mean they separate. So then a nondisjunction is a crazy word for chromosomes don't separate. Okay, so we're going to talk about meiosis later, but basically what it means is the chromosomes tend to stick together, so you end up with too much genetic information in one spot. Okay? All right, so a deletion is just like if you went over to the computer keyboard and you hit the delete button. Part of the chromosome is lost. Okay, it's just gone. Just like if you accidentally hit delete and part of your paper is gone. Okay? An insertion is also called a duplication, and that means a gene is copied twice. Okay, so there's going to be too much genetic information there. And a lot of kids think, well, what's wrong with that? Well, think about if a recipe calls for 1 cup of oil and you put in 2. That could be disastrous. Okay? And inversion, I want you to think of it like a flip. Okay, like if I did a handstand, I would be inverted. Okay, so the genetic info is upside down. Okay, and if you want to use our cake example again, that would be like me baking the cake before I mix it. Okay, things have to be done in a particular order. And then a translocation is the worst one. This is when it is deleted off one chromosome. Okay, and then it tries to fix it, but it attaches it to another. Okay, so in other words, you're actually affecting 2 chromosomes there, and most translocations end up in death. Okay, so let me show you pictures of those. So we're just going to treat these letters, these alphabetic letters, as genes. So if you'll notice, here's the original chromosome, here's the new one. Notice that gene B is gone. Deletion. Okay? All right, insertion, which is also called a duplication. Notice with the new chromosome, gene B is twice. Okay? So that's a duplication or an insertion. An inversion is the chromosome breaks off and then it tries to fix it, but it puts it in there backwards. So instead of this being A, B, C, D, E, F, it's A, E, D, C, B, F. Okay, so all the steps would be backwards then. And here's the translocation, which is the terrible one. Okay, 2 chromosomes, part of both of them are deleted, and notice that this arm of this chromosome is now on this one, and that arm of this chromosome is on this one. Okay, so those are the worst kind, and like if that were a developing baby, it would probably die instantly, probably before the mother even knew she was pregnant. Okay, so all of these things can actually be spotted on karyotypes that we did before. Okay, all right, so let's just review these super quick. Okay, so a frame shift mutation, that's the one where the entire strand shifts. So I like to use that, move, shift. Okay, part of a chromosome is actually repeated. That's a duplication or an insertion. Okay, a single nitrogen base affected is a point mutation. Part of a chromosome is inserted backwards. That would be an inversion. Part of a chromosome is broken off and attached to another one. Remember, that's the deadliest one, a translocation. And then part of the chromosome that is lost is a deletion. Okay? All right, so frame shift or point, and I'm going to give you all a hint here. Do you see how this one has an extra nitrogen base? Let me get off of that. Okay, this one has an extra nitrogen base, and so does this one. Those are your clues that both of these are frame shifts. Okay, and then the middle one is a point mutation because all we did was change one particular thing. And I don't know where my T went, guys. So frame shift, point, frame shift. Okay, and then looking at these, your first clue for this one is that there are 2 chromosomes originally and 2 at the end. So that's obviously a translocation. Okay, this one, the whole center section of the chromosome is gone. So that's a deletion. Okay, and then this one, see how it goes CD, CD? So that is insertion or duplication. And then this last one, this thing spins around and gets put in here backwards. That's an inversion. Okay? All right, so guys, I've got one Google form for you to complete on mutations, and then we're actually done with this unit. We just have a couple days to work on some genetic technology stuff and how we can find criminals and baby daddies and stuff like that, and then we'll be done. Okay? So you guys have a good day. Make sure you call me or email me if you have any questions.